Okay, don't everybody leave. This could be good. So. Okay, so I'm Cenk Sumen. I'm standing in for our CEO, who's a little bit under the weather. He sends his best regards. So if you came for the main act, I'm kind of like the cover band. But I will do my best to present how excited we are at Maxcyte to be pushing in new directions and our partnerships. So the usual disclaimer, with investments, trust professionals. Investors, I'm not one of them, but we have to put this up. Well, this is our attempt to put Maxcyte in a nutshell. It's a 20-year history, so it's hard to capture on one slide, but when I explain this to audiences, what do we do at Maxcyte? I say, look, the cells are little machines. We put them into patients to carry out specific functions. They need a code, they need software. These are molecules, you've heard of DNA, RNA, different editing methods. These have to be transported to the cell. And we're the system that can move this cargo into the cell in a way that doesn't harm the cell. That's a safe, clean approach to recode the cell, essentially. So this works with any cell. It can work down to the level of one cell, and we've gone up to hundreds of billions of cells with one step in 30 minutes. I'll talk more about that. We do have industry-leading performance, and we're always very happy to try our systems with different cells and different processes. We've had steady growth, as most of us have over the past couple of years, as more and more investment has come into cell and gene therapy. And I'll present some data on that as well. We have a lot of collaborations. We've always supported collaborations across industry and academia. We do have early significant investment in the regulatory space. We filed an FDA, confidential FDA master file 20 years ago now, and many of our partners have benefited from this foresight that we put in place. And I'll give some information about the clinical trials, the types of cells. Basically, we have a partnership model, right? So it's not just an instrument or a machine, but it's a partnership over the entire life cycle of the product. And I'll talk more about that because it's an unusual business model, but it brings a lot of value to our partners. So a brief history of Maxcyte, also in one slide, and we attempted to graph here the impact we made on the industry in the y-axis here. So it's just an estimated impact, but you can see our initial foray into cell therapy and then drug discovery as well, our filing of the drug master file, our listing on the Toronto Stock, I'm Canadian, so I said Toronto Stock Exchange, the London Stock Exchange in 2016, our launch of the Expert series of instruments in the tens, our listing on the NASDAQ last year, and our launch of the VLX instrument at the very high end of scale in the industry. This was just 2 weeks ago at the BPI in Boston, which some of us were there. So just some numbers, and some of this is kind of a slide we update every year, and it's very impressive to see the investment in cell and gene therapy. So just pointing out over 1,000 cell and gene modified therapies, many in preclinical development, and just recalling from Tim Hunt's presentation yesterday, over 300 late-stage pivotal trials as well. So we should expect a lot of commercial approvals over the next couple of years. And the focus in 2022, apart from supply chain disruptions and everything else as we come out of the pandemic, has been a lot of interest in other cell types. And I'm sure these cells don't like being called other cell types, but just to look at the origins of the industry, bone marrow transplants, CAR T cells, dendritic cells, now we're really seeing a lot of courageous approaches to use cells that haven't been used before, right? And we have a different view on things like durability, where these cells need to go after we inject them into the patients, even a lot of combination of polymers and cells and organoids. So that's actually one very encouraging development. And both autologous and allogeneic are advancing very well and have a space because in the end, it's the effect on the patient that should decide what gets into the market. Of course, it has to be sustainable and manufacturable, and I'll talk a little bit more about that as well. And rapid growth in targets. So I could put up a lot more genes, but I usually say if your favorite gene is not on this list, please come talk to me and we can see what we can do. A little bit about the partnering and how we bring value. Little less than a year ago, I was a cell therapy developer I was CTO of a small company. We were raising money, trying to take academic process and move it into a space where we can manufacture clinically. So I really understand the pain points. And I've listed some of these pain points as challenges here. On the left, supply chain issues this year, turnaround times, vector supply, capacity, the complexity of edits that we're doing, which is actually great because there's so many new circuits, molecular circuits to build inside cells that might be clinically useful. Regulatory risk that comes with this, and just the inefficiencies of the processes that we're familiar with. So we live this pain with our partners. We have solutions. Part of our approach is to reduce the complexity of cell engineering with one buffer, one system, one set of technologies that can work and can be optimized to each cell type to have very high efficiency and viability. We pioneered flow electroporation. Really proud of this, so we've always taken it to the next level. I'll talk a little bit more about that. And we have very strong regulatory support that helps our partners de-risk their compliance in their march to the market, to commercial approval. A little bit about the expert platform. This is our set of instruments. I don't want to imply that we're just about instruments, but it's always fun to show these, and they're very beautiful and they work very well. High performance instruments. We use one single buffer that's very simple. It doesn't have any additives. And it's useful for GMP compliance. We can go from a few cells to now hundreds of billions, which is really impressive to see in action. I love being in lab to watch this. And we can engineer any cell type, and we're protected by a strong IP portfolio. So our expert platform allows us a unique approach to partnership, and this is a sustained, stable partnership for the long haul, for many years, over the entire product life cycle. Our instruments come with a full warranty, high performance, they're GMP compliant, they can be used in a research use only setting, of course. They're really not designed for academic labs, they're designed for the factories, right, that make the products. 24 CFR Part 11 compliant, network compatible, and compatible and integratable with upstream and downstream technologies. Very proud of our FAS team. We have an awesome team of field application scientists, and a lot of fresh talent has come into the company. Some from actually academic labs, friends that I know in academia, and we're always very happy to mentor these newcomers and help them transition into manufacturing from academic research. We have deep and very technical expertise. I was able to talk to some of our clients, potential customers, and our partners at this meeting today. There's always challenges, and we're very hungry to help our partners solve them. And very strong regulatory support with the company, global regulatory support, and quality systems, compliance, and operational improvements. We're very happy to have celebrated the ribbon-cutting ceremony of our headquarters in Rockville, Maryland. And I invite everyone here, if you're in the Washington, D.C. area, to come by and say hi. It is a really beautiful facility, and we're now have space to expand and to develop our manufacturing systems as well, to be able to handle all this growth. Now, just a few data points about our partnerships. So we've mapped out, we do have 17 partners, SPLs we call them, that have been signed since 2017. And we think that the addressable market is about 50 or so, right? So we have about a third of the market. This is growing. We have 95 plus programs, almost 100 clinical official licenses, and about 15% of these have entered the clinic. I'll show some more data on that in an upcoming slide. So you can see some of our partners here. And it's a very diverse group of companies working on a lot of different cells for a lot of different indications. And it's not just large companies, it's a nice blend of small, medium, and some very large companies as well. So this is a nice chart to see how the clinical trials that use cells that have been engineered by Maxcyte systems are distributed across the space. So let me just take take everyone through this. So the outer rim is basically phase 1, and as these products move through clinical development, they move towards the center. You will note that there is no dot in the center yet. We haven't had a commercial approval of a product using Maxcyte engineered cells, but we're eagerly awaiting this in the next year or so. And it's a nice blend. In pink, you can see autologous therapies, and in green, you can see allogeneic. So it's a very nice mix. It works well with both types of cells. Of course, this is represented, these products have taken many, many years to go through clinical development, so it's heavily represented for some of the early pioneer cell types like CD34s and CAR T cells, but I'm very excited to share that this other cell types and approaches arena is growing rapidly as well. Now, if we look at the types of diseases, again, it's heavily focused towards genetic diseases and blood cancers, liquid cancers essentially, because this is the way the industry has developed. A lot of exciting work on solid tumors. That's one of the holy grails, of course, of the industry. And towards your upper right, just a short list of editing technologies. I think this is interesting. Working our way from zinc finger nucleases to TALENs, RNA-based methods, CRISPR, base editing, which is also sometimes referred to as CRISPR 2.0, ARCUS, and many others. And as I mentioned earlier, we're expecting the first therapy that uses cells engineered by the Maxcyte system to gain commercial approval. So this will be a nice milestone for all of us. Okay, wrapping up, this is my last slide. What's our summary and outlook for the rest of 2022? Maybe I'll just mention some of our achievements. This deck was meant to be presented by our CEO, so it's very immodest. I would never do this, but I really admire our CEO's confidence in me. I hope not to let the team down at all. We added 2 partners this year, Intima Biosciences and LG, so basically expanding our global reach as well. As I mentioned, we completed our move into our new headquarters with a lot of space, and the labs are looking beautiful. And we launched the VLX instrument. I'm very excited about this because I won't get too much into in this talk, but please do find me, talk to me. I've already discussed this with some of the groups here. It can really transform the way products can be selected early on in a process. And when I say products, it could be biologics, it could be anything basically that the cells are used to manufacture, and really extends the range of transient transfection, because we need more and more material these days to be able to decide which product to go forward with. So what's our outlook for the rest of the year? We are launching new process assemblies. These are the consumables that go into the instruments. We're continuing to build out our in-house manufacturing capacity, engaging in automation and integration partnerships, and we're investing in applications and PD labs to enable innovation and to work closely with the partners so we can test out some of these concepts using the same bioreactor, same sensors, same systems that our partner use, to be able to gain more confidence building these processes in lockstep with our partners. And we're in a constant seeking mode for new complementary synergistic technologies that we can use to add to the value that we bring to customers. And I've already spoken to many people here about potential collaborations, so we're always open. And with that, I'll maybe have time for 1 or 2 questions. And I invite you, thank you for your attention, I invite you to come up to me and ask any questions. Thank you very much.